was that film? Outbreak? Dustin Hoffman, was that it? Classic film. Ebola virus, where they used the shit out of this picture, didn't they? It was something like this, where they looked down the microscope and they saw yeah, that was it. They looked down the microscope and then they compared one copy to another. They first of all, they saw an image like this down the microscope, which is complete bullshit. And then they compared it against like an earlier slide, and they were like, the virus is mutating. Hello. Let's do this. Welcome back to the stream. So, today is Thursday, and on Thursdays, I've been doing this series called The Body You Live In. It's a kind of back to basics about human biology and kind of things that go around with that. We mainly talk about molecular and cellular biology, but you know, we venture out a little bit, and tonight we're going to venture out a little bit into the world of viruses. So, change the scene. These guys. So, as I'm sure you all know, this is an electron microscope image of a coronavirus. Thought I'd go all topical, given the pandemic and everything. And yeah, this is effectively what coronaviruses look like. This isn't specifically the SARS-CoV-2 virus. This is another just kind of generic coronavirus from a few years ago. But what's cool about this image is the resolution of this image. So it's in black and white, and it's quite grainy, and I'll tell you why that is in a minute. But you can see that this virus has got a kind of a membrane or a capsule around it, and it's got these little protrusions coming out, which are surface proteins. Now, the reason why it's so grainy and the reason it's in black and white is due to the fact that this is what an electron micrograph image looks like. So when you're taking regular microscope images, where you get nice colourful ones, you are using a light microscope because you can, because what you're looking at is bigger than the wavelength of light, and therefore you get an accurate representation of the image when a wavelength of light bounces off that and into the eyepiece, right? What happens when your subject matter is smaller in size than the wavelength of light? Well, you can't use light. You can't use it to view it. So you have to use something else. And people years and years ago came up with the clever idea of firing electrons at things, which are smaller than, have wavelengths that are smaller than the wavelength of photons. And they can resolve images, and these images kind of look a little bit like this. These viruses are so small, you wouldn't believe it. So I'll go over the size of them in what we're going to go through. But as per usual, we've got the Google Docs out, and just a little structure for tonight. We're going to be looking at, effectively, I'm going to be telling you what viruses are. We'll go through their basic structure, their size. Different types of viruses, which is mainly determined by their genome and their genetic material. So we'll go through something like that as well. We'll talk about their replication, which is really important. And we'll talk about their role in disease, which is going to be kind of familiar to a lot of people. And I've got some examples of 3 different types of viruses that cause different types of diseases, which you should have all heard of. One being influenza, one being HIV, and the other one being Ebola. I didn't want to talk about the COVID-19 causing virus because, frankly, I think everyone's had a bit too much of that. But you know, the majority of what we're going to talk about today applies to that as well. So that applies to that virus as well. So it's not too different. Okay, what are viruses? Well, that is a very, very good question. So, can you call them life forms or not? This is an interesting one, right? So, some people believe that viruses are the smallest or most primitive form of life on the planet. And others would say that actually viruses aren't alive. They are just effectively replicating machines. I personally stand on the latter. I kind of go for them as not really being alive, and that's because of things we're going to talk about. But the main thing is that they can't actually survive and replicate without a host cell to be in. So what we'll definitely say is that viruses, and we'll break out the red, viruses are the smallest form of life, in inverted commas. And not only are they the smallest, but they are the most simple as well. If you want to call them alive, which we're not, but you know, I'm not. You can make your own decision about it. So they are replicating. They're replicating particles, if you like. With a genome. This is where the confusion about life comes in, because they have genetic material. They are the smallest and simplest form of a structure with genetic material, but genetic material is absolutely essential. Replicating particles with a genome. But they do, the biggest thing about it is that they require a host cell. I've realised why I can't type. It's because my microphone is right over my view line to my keyboard, and I can't touch type. So I have to look at the keyboard, and that's why I mess all of this up. But it's one of those things I could lean back and do it. For one day, I'll learn how to touch type, and it'll all be easier. Require a host for replication. This is key. Absolutely key. Alright, so let's jump. I will come back to basic structure, but let's talk about size first, because size matters. We have, I've managed to find a nice little Tetris-esque type diagram that seems to be quite accurate, right? So they list some really small stuff on here, but the thing you need to look at is the fact that it's got a millimeter up top. So then we have a reference point. We have 1 millimeter, okay, and this is in diameter. So at 1 millimeter, you are finding the very, very largest things called eukaryotic cells. So humans have eukaryotic cells. We are eukaryotes, which means that we have a certain cell type within us. Our structure of our cells is in a certain way. We haven't really gone through the differences in those kinds of cell types, but we might do in a future video. So we have eukaryotes, and then smaller than that, we have prokaryotes with a little bit of overlap, and then we have the viruses. So viruses occupy this kind of less than 1 micron, down to times 10 to the minus 8. So what that actually translates to in kind of real world terms, is try and give you this point of reference. So we're going to have 1 millimeter actually. Yeah, okay, so we're at 1 millimeter, because you can reference that, you know what that is, right? So a micrometer, a micron as they call them, so micrometer or a micron, is equal to 1/1000th of a millimeter. So that means if you took a millimeter, you divide it up into 1,000 pieces, one of those pieces would be a micrometer. Okay. A nanometer, abbreviated to NM, or nanometer, is again 1/1000th of a micrometer. So you see we're going in thousands, right? So a micron is often using the Greek letter mu, which we'll just write as U. A nanometer is 1/1000th of a micrometer, which makes it 1/1 millionth of a millimeter. Okay. And 1 millimeter is 1/100th. No, 1/1000th of a meter, which is the standard empirical unit. Which is the reason why we're going down to 10 to the 8 and 9. Down to the minute, down to this kind of scale. So viruses are around, they vary in size, but they are around 20 to 300 nanometers. Okay, so that's pretty damn small. Okay, that is shorter than the average wavelength of visible light. So it's tiny. It's really, really small. And if you compare that to, say, an animal cell, like a human cell, so human cell is about, from my notes that I've got here, can be up to around, it's 40 to 100 micrometers across, and a bacteria is somewhere in between, with about 200 to 2000 nanometers. So let's put them into the same units just so we can understand where they sit. 40 micrometers is 40,000 nanometers to 100,000 nanometers. So you can see that viruses are smaller than bacteria, and they are smaller than human cells. Massively smaller. Just to give you an idea of scale and size of the world around us and all these agents that are causing us all this ill health. They are very, very small. And right, so now we have to talk about that basic structure. So I've effectively stolen this from Wikipedia because they have good pictures. But you can think of a virus, effectively, in the majority of cases, as a piece of genetic material, either RNA or DNA. If you don't know what RNA and DNA are, I have a previous video or previous stream with that kind of detailed. That is usually coiled within some sort of protein subunit or subunits, and that's it, really. Sometimes they might have a membrane around the outside of them, but not always. And that is really the simplistic level of viruses. So the basic structure is it's just basic, right? Very, very basic. You have some genetic material, and you have, so you can say usually it's within a protein structure. Protein, sometimes it's called the capsid. Possibly within a membrane, depending on the virus. But it's these bits that are common. Bang that in red. And the genome can be RNA or DNA. Now, there's different forms that you get the genome in. Some of the genomes can be what's called double-stranded or single-stranded. So we'll put that here. So just for reference, the standard DNA helix that we have in our bodies, that structure is double-stranded because it's a double-stranded. It's basically a ladder that's been twisted. Two rungs of a ladder, two arms of a ladder with rungs going across it, and it's been twisted. That's double-stranded DNA. You can also get single-stranded RNA that's coiled in a different fashion, and you can get single-stranded RNA and double-stranded RNA. So we'll say single-stranded stroke, double-stranded, again single-stranded. Slight different flavors on the fact that it's genetic material. Now, what I'm not going to go into on this, and if you're into virology, you might already know this, is that actually there's another extra layer of complexity on the genomes, which is called positive sense or negative sense RNA and DNA genomes. That's effectively the way it's read. Not going to go into it too much today, if at all, because it's a little bit too advanced, I think, for this kind of very basic level understanding. So this is an example of a virus. I mentioned about a capsid, a protein box that the genome sits in. This is an interesting one, right? So this is a type of virus called an adenovirus, and it has what was theorized to be an icosahedral capsid. So an icosahedron is anyone that plays Dungeons and Dragons will know that it is a 20-sided shape. And what they've done here is they've actually got some electron microscope images. And if you look very, very carefully, you can see that there is actually this shape that resembles an icosahedron viewed from a single plane. And they've overlaid a shape over the top of that goes to show that, you know, it's quite a complex protein structure. These capsids, each side of these might be made up of, say, 3 or 4 different proteins, all assembled. But generally, that's quite a simple structure. And I think what the take home of what I'm going to talk about today is the fact that viruses are very simple, but they do lots of detrimental things to humans. So yeah, they have different shaped capsids. So let's say capsids, different shapes and sizes. Shapes, sizes, and sides, I guess you would say. Let's put icosa, so you can't type at all. Is that how you spell icosahedral? Google seems to think so. Fine by me. Okay, so what are viruses? They are the smallest, simplest form of life. They are replicating particles with a genome that require a host cell for replication. They have a very basic structure, genetic material within a protein capsid, possibly within a membrane as well. The genomes are usually double-stranded, single-stranded RNA or DNA. And capsids are of different shapes and sizes, icosahedral being a very interesting and quite common shape. Oh, something else to note up here with what are viruses. Viruses are not unique for infecting mammalian cells or human cells or even animal cells. You can actually get viruses that infect bacteria as well. They are called bacteriophages or bacteriophages if you pronounce it like that. So you can say they can infect eukaryotic cells, and that would be like animals, e.g. animals. But you could also get prokaryotic cells being infected by viruses. Infected by phages. And actually, what I'll do is I'll show you a picture of a bacteriophage because it is... I can't spell it at all tonight. Bacteriophages, you may have seen, they look like kind of ants in their structure. They're usually slightly different. Same principles are adhered to. The idea of a genetic material within a protein capsid. Usually bacteriophages have these structures that they use to get their genetic material inside of their bacterial cell hosts. The reason for this is that bacteria occasionally have cell walls and thicker structures to get through. But we're not going to go into that. We just need to know that you get bacteria that get infected with viruses as well. Cool. Okay, so we're going to touch on the types of virus you can get. Aside from kind of standard viruses and phages, which we've talked about, we need to talk about the genomes. So, as I mentioned, you get RNA versus DNA genomes. Where have I put the... And this is what this is going to do. The genome structure is going to tie in quite a lot with the replication, which we're going to talk about. So the genome is going to be single-stranded RNA or double-stranded. It's also going to... This is also going to tie into the role of the immune system in detecting viruses in disease as well, because in our bodies we have double-stranded DNA, and it's not normal in humans to have single-stranded DNA, which means when a virus comes in that has single-stranded DNA, it's detected as foreign because we don't normally have single-stranded DNA. Similarly, we don't normally have double-stranded RNA. Most of our RNA is single-stranded. Okay, they have a virus genome. The genome can vary massively in size as well. So I've said that they're simple, but you can get some very large viruses. So I think the tobacco mosaic virus is huge, if I remember rightly. So this is a virus that infects plants. You get viruses that infect plants. And this has one of these structures. This is where that image came from. Long helical structures. This is the virus, by the way. You know, it's effectively a long pipe with genetic information in the middle. Do we have anything on the genome? Let's see. Oh, there is a section on the genome. Genome. There you go. Right. Consists of 6.3 to 6.5 kilobase single-stranded RNA. That's 6,000 bases, which actually isn't that much in the grand scheme of things. You know, we've got a few billion bases, so having a few thousand is not too much. They are very simple, and usually they, well, in this case for the tobacco mosaic virus, it only has 4 genes. And if you remember from previous videos, a gene is a region of DNA that encodes for a protein. A region of genetic material in this case that codes for a protein. In this case, it's RNA. It has a replicase, an RNA helicase, an RNA-dependent RNA polymerase, and a so-called movement protein and a capsid protein. Usually, so the viral genome will always contain a capsid protein. That's the structure it effectively lives in. So let's add that to the list. Usually it's a short genome. And we're talking, you know, maybe let's say 2 to 6. No, but I don't know what the range of genomes are, so I'm not going to try and guess. Usually they can vary massively. They have a short genome. What I will say is that usually what will happen is you'll get a single polypeptide being produced from that genome that will then get cut up into its respective proteins. This is different to how a lot of mammalian proteins work, where they're actually set pieces of a gene that are set pieces of a protein that are produced in full, like one protein is produced, rather than this polyprotein that's produced. So so polyproteins are produced and they will contain a capsid protein, and that's for during replication. You're going to need, if you want to make more viruses, then you're going to need to make more capsids, or the cells are going to need to make more capsids. They may have proteins that enable their own replication. They may have replicases in there. They may not have replicases in there. They might be a virus with such a genome that actually the host machinery can do everything that it needs to do in terms of replicating it. But some viruses have certain genomes that need to be processed in order for the cell to then do a bit more processing. So they might have replicases. What else might they have? They may have, in terms of retroviruses, they might have integrases. And we'll come on to integrases and retroviruses when we talk about HIV later. It's a classic retrovirus. They might have integrases, they might have helicases, which they use to unwind their genome in order to allow access, replication, and for protein production. And we'll leave it there in terms of they're probably going to, other genomes will have different proteins encoded within them as well, depending on what type of virus it is. Something we should talk about, though, is some viruses have segmented genomes, and flu is a classic example of this. So could be segmented genome, and we'll talk about this a bit more with flu. But the reason why that's important is because different segments from different viruses, so you've got flu virus A and flu virus B, which are very different. If they end up in the same cell together, they could swap components of their genome, and you could get flu virus C coming out. Effectively, you can mix and match, which gives rise to a large amount of genetic variation within viruses. Excuse me 1 second. Okay, so we have got types of virus and the genome. Lots of different types of viruses exist. Plants, animals, bacteria, viruses. Replication, right. So replication really depends on the genome. So that's why I wanted to take you through the little bits about the genome first. Depends on the genome of the virus. It also depends on the host cell that it's infected. So I mentioned that they can't replicate outside of a host. So when I'm saying a host, I'm talking about, a cell in our bodies that is harboring a virus. So in the case of the SARS-CoV-2 virus, it will be the epithelial cells within our lungs and our throats and our nose and the back of our mouth. Mainly, other cell types are applicable. So it does depend on the host cell. So replication is dependent on the host cell. Viruses cannot replicate outside of host cells at all. So virus dependent on genome, dependent on host cell. So let's take an example of a single-stranded RNA virus, because it's quite an interesting little thing, literally. So, e.g. virus. Now we know that I've shown this picture many times, but we'll show it up again. It's called the central dogma of molecular biology, and it's this idea that you get. Let's go on to this one. It's a bit easier, a bit simpler. It's this idea that you have in your body regularly in your cells. Nothing to do with viruses. You have DNA. DNA is converted into RNA. It's transcribed into RNA, and then the RNA is translated into protein. Now, if you are infected with a virus that has a single-stranded RNA genome, you're already at this step, this middle RNA step for the virus. The virus is already here. So what happens is that your cell doesn't know the difference between human RNA and virus RNA. So it just sees the RNA and it goes, oh, I'm going going to make some protein out of that. Okay, and so what happens is that, you know, host cell effectively can't tell the difference between viral RNA, specifically single-stranded RNA, and its own mRNA. And mRNA is the RNA that we showed you up here that it uses to make proteins. So it starts converting or translating mRNA into protein, but that mRNA actually is, that protein that's produced is actually the viral capsid, usually proteins, because they're usually the most expressed or the most produced. So the host starts making viral proteins. Okay, so what happens at the same time? So you've got a host that's making a host cell that's making viral proteins. You've also got what might have happened is that the virus brought with it its own replicating enzymes. So replicases here. Okay, so at the same time, the virus is replicating its genome. So you can see what's happening here, right? So your virus is replicating its genome, but its genome itself is what's being translated into protein. So you're just going to get more and more virus proteins being produced. It's going to completely compound, and it's going to overwhelm the cell and the cell's machinery. It's just going to get more and more and more until effectively that cell will only be making viral proteins, in which case it's probably going to die, which manifests itself in disease later on. So what happens when, I mean, these viral proteins, so it's making a capsid protein, they can self-assemble. So the capsid proteins self-assemble, and contained within those viral proteins is likely to be the viral mRNA, which is effectively a new virus. And then you get so much what's called virion. So virion is like a singular term for a virus particle. Virion production effectively overwhelms the cell, and either it dies, the host dies, releasing virus, or in other cases it bursts, releasing virus. And that's effectively how viruses spread. So you can see why people are kind of a little bit apprehensive about saying that the viruses are actually living things, because there's no choice in what they do. They go in, they replicate, they overwhelm the cell's machinery, they just keep replicating and replicating and then punch out through. They have a capsid protein that will self-assemble, and therefore in that self-assembly will be more genome, which will then get released. So what's an interesting thing about mRNA? What's an interesting thing about viral RNA and viral RNA genomes in this instance is that you don't actually need a virus to infect a cell. You can just inject, for lack of a better term, this RNA molecule into the cell. More than one, I would imagine, but you know, a proportion of RNA molecules into the cell. Leave it for a certain amount of time and you'll get fully formed virus particles out the other side. So effectively the RNA that you're injecting is virulent. It produces viruses. So you can get viruses from just genetic material. No other life form you can do that with. and therefore you can ask the question: is a virus a life form? Don't know. So, giving you an example about the replication, there are obviously other forms of replication you can get if you have DNA viruses. They replicate slightly differently with different enzymes, and in fact, some DNA viruses can utilize human DNA replicators, so they don't have to bring their own like RNA viruses do. And sometimes the the viral genome might be what's called negative sense or positive sense, which basically means the direction that it's read. I've mentioned in a previous video that machinery in cells only really reads DNA and RNA molecules in a certain direction. And so sometimes the encoding is in the other direction, so they have to make a copy of it in the right direction and then use that as a template to start building virus particles. But we don't have to go into that too much. So let's get on to some of the examples that we've got. So we've got role in disease. Obviously, we all know that viruses are responsible for a lot of diseases. We're in the midst of a pandemic right now. But every year we have a seasonal flu epidemic. We have the HIV pandemic, and which has been around since the 1980s. And we also have another example is Ebola, which is a virus which causes an absolutely horrific disease that is born out of a virus as well, and they all behave slightly differently. So let's go through that. Let's bang the mic arm as well. Okay, so flu is one of the most well-studied viruses going. So influenza is its official name. Flu, I've got a few notes down here that I'm properly cheating with. Okay, so flu is an interesting one because of its genome and because of its variation that you get. And let's talk about this. So this is flu. Different electron microscope image of flu. TEM, transmission electron micrograph. This is the 1918 influenza virions that were collected from supernatants of an infected Madin-Darby kidney cells type of cell 18 hours after infection. And you can see that they've got this kind of, again, the structure with proteins on the outside. And within here, what you can't see is the genetic material that's inside them. But what you can see from this is that they're not the classic icosahedral shape that we've looked at earlier on. So what I wanted to point out with flu is because we've had a few flu pandemics over the last few years, swine flu being one of them, avian flu being another one. And this just refers to the first instance of zoonotic transfer, so transfer from animals into humans. Influenza can infect a load of different animals. They infect birds. It infects birds very readily. It doesn't actually cause a disease in birds, I don't believe. And but when those viruses transition over to being able to infect humans, then we have a little bit of a different response, and we get into trouble. We end up being very sick. But what's cool about flu? So let's see if we can bring up. What I want to show you is this. Yes, here we go. So let's just zoom into this. No, I don't care about the bottom bit. Okay, so flu is it's a virus that has a capsid, like most viruses, but it also has a lipid envelope, so it has a membrane around the outside of it, and that gives rise to its kind of less structured shape, shall we say. not like an icosahedron or anything like that. It also has, very importantly, it has an RNA virus. It has 2 major surface proteins. One's called hemagglutinin and the other one's called a neuraminidase. Try saying that after a few. And it's these proteins that give rise to the names, things like H1N1. Hey Jimmy Dreambeard, how's it going, man? I'm good, thanks. Just talking about viruses, how are you? So it's hemagglutinin. Let's see if Google can cope with my shitty spelling of this. Oh, it's got it. Oh no, it hasn't got it. There it is. Hemagglutinin and neuraminidase. That's going to be wrong spelling as well. Yeah, it is. 2 for 2. There we go. This gives rise to the H1 or HX and X. And this is the type of these proteins that they have on the outside. And it's just an identifier. So it's an identifier of the different types of virus particle. Another thing that is quite interesting about flu virus is the fact that it has what's called a segmented genome, right? So I think it has, is it 8 separate pieces? Separate segments of its genome? I think it is. Let's have a look. Where's the genome? I'm pretty sure it's 8. Type of virus, subtypes, nomenclature. I did have a nice picture of it, but I think I lost it when I shut down my computer. This one actually, this highlights what we're talking about. So it has this segmented genome. It's actually 1, 2, 3, 4. Oh, 1, 2, 3, 4, 5, 6, more than 8 probably. And the reason why that's clinically important for us is that you can have, in this example, say like a highly pathogenic avian strain with its 12 segments, and you can have a human strain that's quite weak in comparison. But if both of these viruses infect the same cell and these genomes mix, you get viruses coming out the other end that have a combination of avian and human genome segments. And that's something that the immune system's never seen before, and it can potentially be highly virulent. And that's why you end up with these epidemics and then pandemics that kind of spread across the entire world, like avian flu, swine flu is exactly the same thing that happened. So it was a mixture of different strains of a flu virus that occurred usually within the animal species rather than within the human, that then could jump to humans. When am I going to fix it so we can go back to the cricket? Oh, I'm working on it, mate. I'm working on it. Though the cricket's going to be washed out this week. Mark my words. It's going to be a shit show. We're going to be on the edge of winning, and then it'll be a washout for days 3, 4 and 5. Okay. As a segmented genome, and you can get mixing of this genome to give rise to new species. Well, it's not species technically, it's subtypes of virus. And these can be, the new viruses can be highly pathogenic. It's worth pointing out though, this is not a certainty, right? So they can be highly pathogenic, but they are actually the most likely outcome is that they're not pathogenic, or any more pathogenic, really less pathogenic. It just so happens that you only need one combination to be right, to be kind of correct, and then you're in a world of trouble because of the rate at which the virus can replicate and spread. Oh, you appreciate it? Thanks, mate. Better people than me are working on the current pandemic. I can guarantee you that. So something you may have heard of, that this process is called antigenic shift. Right? And it's because it's a massive shift in the genome. You also get something called antigenic drift in flu viruses and a lot of other viruses, which is where the genome changes subtly, a base pair here, a base pair there, part of a protein removed, part of a protein added or coded for, and that gives rise to smaller changes in the virus proteins and the genome. So smaller changes in proteins. So it's smaller changes in the genome gives rise to smaller gives rise to smaller changes in the proteins, especially the surface proteins, right? Surface proteins, the H and the N. This is called antigenic, if I can spell it, drift. An antigenic drift is the main reason why we need a new flu vaccine every year. Because what happens is, over the course of a year, the H and the N proteins shift. They drift. Sorry, I should use the proper terminology. They drift slightly, i.e. they slightly change in their structure, and it renders the immune response that we've generated from the last vaccine to be useless, effectively. And we need a new vaccine that's based specifically on those antigens in order to combat the virus. Doubt it, you're A1, Jimpy. Thanks, mate. Thanks, mate. Like and subscribe. And this is the reason for new vaccine every year. So when people ask you, well, how come when the anti-vaxxers ask you, well, what, the vaccines don't work because you need a new flu vaccine every year, you can say it's due to antigenic drift, and then you can send them to my channel on YouTube to watch the video, where they will go, what the hell is this guy talking about? I genuinely have an anti-vaxxer on my Facebook feed, and he provides me with no lack of opportunity for correction. Okay, so we've done a little bit on flu. Let's talk a little bit about HIV. HIV, I mean, everyone's heard of HIV, causes AIDS. HIV stands for human immunodeficiency virus. AIDS stands for acquired immunodeficiency syndrome. And the reason that your immune system effectively goes to shit when you have HIV infection is because it targets cells called CD4 T cells. Now, I'm going to do a stream and a video on the immune system and the importance of CD4 T cells in the future. Might even be the next one. CD4 T cells are probably the most important cells in your immune system, and this virus directly infects your CD4 T cells, and upon infection your cells respond to that infection, and one of the responses to that infection is to tell the immune system that I'm infected with a virus, and then your other immune cells come along. They don't discriminate against any type of cell. If they see it's infected with the virus, they kill it. So, causes AIDS. Let's put this right, by targeting cells called CD4 T cells, and then CD4 T cells are targeted by the immune system for death. And the result is a weakened immune system. Yeah, that will convince them, I'm sure. Weakened immune system, and then sadly, people that actually have HIV/AIDS that are not when it's not under control, they eventually succumb to other opportunistic infections and certain cancers that the immune system would. Yeah, sad panda. Is there a sad? Let's not do a sad emoji. That's pretty dark. And they will succumb to opportunistic infections. And cancers, which the body would normally deal with. So, pneumonias, respiratory viral infections, respiratory bacterial infections in HIV patients can be incredibly deadly when they wouldn't normally be to someone that wasn't infected with this. So you might have heard, I don't know, that HIV. You could be infected with HIV and not know it for like 20 odd years. That's what makes it quite scary and hard to detect. So it has something called latency. So viral latency. So viral latency effectively means that you're infected with something, but it doesn't manifest itself in any symptoms until later on. A classical latent infection is things like TB. You can have something called latent TB, where you are infected with TB but you don't know about it, and then eventually at some point it triggers, and then it starts growing again, and then you're in a whole world of trouble. Viral latency in HIV's case is caused by the fact that it's a retrovirus that integrates into your genome. So it's an RNA virus, right? But let's use the terminology. It's an RNA retrovirus. Now, retroviruses, what they do is they convert RNA into their RNA genome into DNA, right? So RNA genome into a DNA genome, and then what they do is they integrate that DNA genome into your own genome of your cells, into the host, which means it's incredibly hard to find. Kind of hides in the genome. That's what Bender from Futurama has instead of DNA. His robo DNA or RNA. Yes, good reference. Love Futurama. Integrates into the host. So it integrates into the host cells. It can lay dormant in the host cells, not producing any viruses, but every time your cells divide, the viral DNA divides as well because it's in there. And then at some point, it may get activated to start producing virus particles. So that's the idea of a retrovirus. Interestingly, we have kind of scientists have hacked retroviruses, right? So HIV is the family of viruses called an adenovirus, and if you read up into any kind of gene therapy, you might have heard of something called an adenoviral vector. And effectively what some very, very clever people have done is they've taken like the shell of HIV effectively, and they've put, instead of having the HIV genome in there, what they've got is they've got this this DNA this construct, this genetic construct that it's designed to be integrated into your genome, and you can do something called cell therapy with that. So if you have a defective gene in your body, say you have a genetic disease, and you want to treat that, what you can do is you can put the gene, like a normal copy of a gene, back into your cells using a fake HIV virus. It's called an adenoviral vector. It's very, very clever. It also means that you can target that to certain cell types. If you know what cell type the adenovirus prefers to infect, you can target that gene of interest, that gene therapy, into the cells that you want to target that are defective. That's how gene therapy works, and it's fascinating stuff. All hacked out of a HIV virus. Probably wasn't an HIV virus, to be honest. I don't know where it actually came from, but it's definitely the same family of viruses. It's completely safe though. There's no chance of developing HIV or anything like that. So finally, we have the scary one, which is Ebola. So Ebola is actually called the Zaire virus, or the Zaire Ebola virus, and it causes something called hemorrhagic fever, which you don't want. You don't want. It's another RNA virus, and it's incredibly infectious. So I mean, the structure is incredibly simple. It's not any integrases or anything like that. It's got this genome here that forms, I think it's a single polyprotein. It's a very, very short virus. The polyadenylated viral genome codes for 7 non-structural proteins and 7 structural proteins and 1 non-structural protein. So 7 of these proteins that are produced are just to make the capsid proteins that encompass it. 1 is an RNA polymerase, that's to replicate the genome, because we don't have RNA polymerases in our cells, so it takes it with it, makes it itself. Replication. Being acellular, viruses such as Ebola cannot replicate through any type of cell division. Rather, they use a combination of host and virally encoded enzymes. Basically, they hijack the host's machinery. So it's got a classical structure. So it's got this kind of looped capsid protein, which is very, very telling. You've probably seen it. I mean, I think that what was that film? Outbreak? Dustin Hoffman, was that it? Classic film. Ebola virus, where they used the shit out of this picture, didn't they? It was something like this, where they looked down the microscope and they saw yeah, that was it. They looked down the microscope and then they compared one copy to another. They first of all, they saw an image like this down the microscope, which is complete bullshit. And then they compared it against like an earlier slide, and they were like, the virus is mutating. And it's complete bogus, because this has to be done with an electron microscope. You don't have like it. You can't do this in like a field hospital somewhere. This electron microscopes are big. You have to process the data. You certainly can't see if something's mutated just by the shape of it, because you don't get that kind of resolution. You need to check the genome. Bad films. Making all the good stuff like pandas and polar bears extinct, yet this narky wanga is still running around. How come? Sorry, I missed that. How come we're making all the good stuff like pandas running extinct? Yeah, that famous virus. That's the one. Jim, you're going to hate Jurassic Park. What are you talking about, mate? That's real. That's real. Anything with Jeff Goldblum is real. Seen Thor Ragnarok? Actually, you know what? That film, Jurassic Park, the whole thing with the amber and the mosquito is not beyond the realms of possibilities. this, yeah, I think that the most random bit of molecular biology in that film was this whole thing about the dinosaurs changing sex, and because they had frog DNA in there or something. That was weird. That was weird. But whole getting like DNA out of, intact out of like preserved samples, we can do that. I mean, DNA is actually quite stable. It's more likely to be like frozen, like woolly mammoth style, than amber, because amber's I imagine quite a warm environment, and that might cause degradation. But yeah. Jeff Goldblum? Is he? Yeah, of course. Is he in? Did you say it's Jeff Goldblum in Jurassic Park? Jesus Christ, son. You do need to go and rewatch that. That's unbelievable. Got the best line in the film. Right. We're getting on to the hour now, so I do want to kind of wrap this up, although we might end up having an argument. Oh, in Thor Ragnarok, no, he's obviously in Jurassic Park, you numpty. Well, I don't know, you need to specify this kind of stuff, James. It's he's in Ragnarok. He plays the Collector. He plays the guy that kind of melts people with his staff. He's got like a killer goatee. Basically, he's the one that imprisons Hulk and Thor to fight. Very, very cool. All right, so I mean, there's not a lot to actually say on Ebola, except for the fact that you've got we kind of want to talk about hemorrhagic fever. If you want some gory pictures, I'm sure that there are some gory pictures not on the Wikipedia site, thankfully. But effectively, the Ebola virus gets in through bodily fluid transfer, and then will basically infect almost every cell going inside your body. And the reason why it causes hemorrhagic fever is because your body, for lack of a better term, your tissues kind of disintegrate because they're so full of viruses that the connective tissue kind of fails and your body leaks and you hemorrhage and you die. It's not just Ebolas that cause this. Ebola viruses that cause this. There's obviously some other ones I'm reading up here called, I won't even try and pronounce them. Signs and symptoms include fever and bleeding. Ooh. Yeah. So, by the size of viruses that we've gone over, and the fact that they're absolutely tiny, you can see how easy it would be to kind of contract them. I mean, you don't need many viruses to actually infect. It will be different for different viral species, but generally they can be quite low. I think that some of the numbers that are touted is that, like hepatitis C virus, for example, you only need actually 20 virus particles, which if they are smaller than the wavelength of light, then it could be quite easy to actually contract that kind of stuff. So that's why vaccines and screening and track and trace in the coronavirus age is actually really, really important. Fair dues, I'll put it on the watch list. Thanks, mate. It's a great film. There's going to be a new Thor out soon with Taika Waititi directing again, so it could be quite funny. All right, so let's wrap this up. We've been prattling on for a bit. So what are viruses? They are the smallest, simplest form of life, in inverted commas. Are they living? Are they not? Very, very difficult to tell. I don't personally think that they are, because they require a host cell for replication, whereas no other life kind of does. There are some bacterial species that can't really reproduce without a host, and some parasites as well. But if you're getting down to the cellular level, I wouldn't consider viruses to be living. They're more likely, I would consider them to be replicating particles that have a genome. And partly because you can just infect a cell with a genome, it's like a naked genome, no virus or anything like that, and you'll get viruses out the other side. So probably not life. But on the border, viruses basically infect all different types of cells. They will infect bacterial cells, they will infect plant cells, mammalian cells, eukaryotic cells, you name it, there'll be a virus that can infect it. Their basic structure is effectively they are genetic material with a protein coating called a capsid. Sometimes they have a membrane around the outside as well. Those genomes are RNA or DNA structured, and the capsids come in all different shapes and sizes. A classical one is the icosahedral shape, 20-sided, but you don't always have that structure. As you can see, flu didn't have that structure, but Zaire virus didn't have that structure. Size, they are very, 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 very small, 20 to 300 nanometers across is what they range in, which is ludicrously small compared to, say, a human cell, which is, about 1,000 times, can be 1,000 times larger than that. And bacteria is somewhere in the middle. So the types of viruses is determined by the genome itself. That genome generally it's very short, it's very simple, it contains the structural proteins, coding for the structural proteins, and sometimes replicases and integrases and helicases depending on what type of genome it is. And it can be a segmented genome in the case of flu, as in the case of flu. And that segmented genome can kind of chop and change with other virus particles, giving rise to new species. Replication depends on a host genome. Replication depends on the host cell in order to replicate its genome. That's a given. It hijacks the cell's machinery to just keep producing virus particles and virus genome over and over and over again. Then you get reassembly of virus particles inside a new capsid that burst out of the cell or are released out of the cell, or sometimes cells are killed off by the immune system releasing the viruses, and so they can go on and infect other cells. Multiple roles in disease. This is just a few things that we've decided to look at, just a couple. Influenza. Biggest thing about influenza is it has the segmented genome that gives rise to antigenic shift when you chop and change this. But the surface proteins of flu can drift in their sequence, in their genetic sequence, and that gives rise to the idea that we actually need a new vaccine every year, because our immune response can't recognize the drifted proteins and therefore can't deal with the virus effectively. And HIV is a great example of a retrovirus that integrates its genome into our own genome and therefore can lay dormant for years. And it's another example of a virus that causes the immune system to kill off a certain cell type, giving rise to downstream disease, which is really, really bad. So I'm going to wrap it up there. I'm going to thank everyone on the stream for watching. Jimmy Dreambeard, thank you for the Jurassic Park chat. I am going to go and have to watch that film because I need my Sam Neill fix. I haven't had that in a while, not since Event Horizon. That is truly his best work. That was really informative, mate. Thank you, mate. I appreciate it. Unfortunately, you're going to have to deal with not having me around for a week because I'm going back home. I'm going back home to visit family for a week, taking a full week off, streaming, work, not doing anything. I was hoping it was going to be sunny and I can work on my tan so that I don't look like the background that I have behind me. But it's going to be raining next week, so, I'll probably just come back as bored and miserable as I am already. But yeah, I'm not going to be around for a week. Still doing the 2 uploads per week, splitting up the streams into videos and putting them on the YouTube channel, so you can go and check that out.